All right, thank you very much. So I'll complete the trifecta of Lake Lab talks today. And I'm going to be talking about polycyclic aromatic hydrocarbons, or PAHs for short. So what are PAHs? They're these things. They are any combination of aromatic hydrocarbon rings that you can think of. So big round PAHs, long linear ones, crazy branch chains, any combination you can think of is a PAH. It's a broad class of molecules and has some really interesting features that we think are very useful for astrophysics. Mainly it's this conjugated pi system gives it a ton of stability. So you can ionize the heck out of it, singly, doubly, triply ionize it. You can add electrons to the system. You can take every single hydrogen off the edge of this ring. You can take every single hydrogen and give it a second one, and they're completely stable, very happy, and will continue to just float in space. They're also highly photostable, so they can withstand extremely high UV flux without dissociating. And you can also do a lot of functionalization. So you can start adding nitrogens into the ring. It's not terribly difficult to add sort of carboxylic acids or ketones or alcohols to the edge of these things. So there's a lot of really cool chemistry that goes on with these. But why we care about them for astronomy and astrophysics and astrochemistry is these things, UIRs. This is one of the longest standing mysteries in astronomy, is these set of near and mid-infrared bands that are seen towards a plethora of sources. And the really interesting thing is they have to be coming from some kind of sp2 aromatic hydrocarbon. The CH stretches and CC stretches all line up perfectly with it. But the weird thing is they don't match up with any individual spectra. You can't take a lab spectra of a molecule and go and compare it and see perfect matchup towards most sources. So what you have to invoke is this weird mix of some kind of big aromatic class of molecules. And so the obvious answer is it's PAHs. But the problem is we've never identified any individual PAH. Also, this is infrared emissions. So any place that's cold or dusty and obscured is very hard to observe. So there's this big gap. We can't identify any individual PAHs. We can't see the size distributions. It's very difficult to tell how functionalized they are. And there's a lot of places we can't naturally look for them. So it's a big ongoing problem in astrophysics. But it's not just a neat problem, it's a big problem. If you look at M81, this is a Hubble image with a Spitzer map overlaid on top of it. And this the red emission here is all PAHs. So if you work through the math, if the UIRs really are coming from PAHs, something like 20% of all the carbon, and for the biologists, it's reduced carbon. 20% of all the carbon in the universe has to be tied up in whatever is emitting these UIRs. Not just that, something like 10% of all the power emitted by our galaxy is mediated through these things. So it's not just a cool little problem, it's actually a really big and interesting problem in astronomy. And the other really cool thing is some of the suggestions are that these things are synthesized in the stellar atmospheres of carbon stars. They then get ejected into space, they stick with the molecular cloud and condense with it all the way down to a protoplanetary disk. And then this carbon gets incorporated into the planets, meteorites, comets in the planetary system. So it's something like 20% of the carbon that's going to be accreted into the planets and also into meteorites and comets that can then bombard the planets as the system evolves. So it really is universal. It sticks with it all the way through the planetary evolution system. So the cool thing for astrobiology is if you go back to something Brett talked about, which is the Murchison meteorite, and you take it and you just grind up an organic sample, you extract the organic fraction, and then you dissolve it in water with fluorescent dye, what you get is a spontaneous formation of these vesicles. And they truly are vesicles. They can sequester the fluorescent dye, or they can be hollow. So it means they're porous. They're not that porous. They can actually segregate the aqueous matter. And the really cool thing about it is it's completely spontaneous. So what this is amphiphilic molecules. So polar head and an apolar tail in the molecule are self-assembling together, and it's just spontaneous. It's on something that's being delivered to Earth, and it's completely abiotic. So I think that's really cool. And so one of the things, if you look into Murchison, one of the big constituents is aromatic hydrocarbons and things like phenol. So functionalized aromatic hydrocarbons are one of the big parts of this. And it's nothing like the carboxylic acids, but it is actually a decent fraction of the organic matter found in Murchison. So there's a lot of organic PAHs, some of which are functionalized and in fact amphiphilic, that are stuck in these things. So one of the really cool experiments somebody did was, this is Murchison on the right, and this is a mixture of organic matter that came from irradiation of ices. So what they did, they take an ice, you put PAHs in the ice, and you blast it with UV light, something like you would find around a star, and what you get out is functionalized PAHs, so ketones, alcohols, ethers, things like that. And when you dissolve them in water in the same way you did with Murchison extract, you get self-assembled little vesicles. So the really cool thing is now you can make something that looks a lot like a cell membrane. So they self-assemble and completely segregate. And the really cool thing for biology is, well, if you look at life on Earth, basically everything has a cell membrane. It's such a massive evolutionary advantage that once you develop it, you never go back. It maintains the chemical gradients, concentration, and gives you protection from the external environment. So it's such a huge evolutionary advantage. Once you make it, you never go back. And the question has always been, when you start to make life on Earth, how do you do it? What's the first step? And at some point, you have to make a cell membrane. The question is, when did that happen in the process of making sort of a self-replicating system? And the neat thing here is that one of the ways you can do this is completely abiotically. You don't have to mess with small molecule synthesis on an early Earth. You can directly deposit a self-assembling system that looks just like, or very much like, a primitive cell as the Earth is forming. So it's a really cool idea, and PAHs are probably one of the big contributors to doing this. So pretty excited about it. But like I said, we've never identified a single PAH, and we'd like to know a lot more about what's going on with these. So the big trick is to go and do astronomy to identify these and to try to identify individual molecules. So Marco already kind of took you through the terahertz spectrum. That's where we like to work. And one of the nice things is, as Marco was explaining, these big polyatomic vibrations are completely specific to the molecule that's doing it. So it's not just the CH stretch of an aromatic species of some kind. Now it's the vibration of a specific PAH. So we'd like to go and do observations of these things. And our favorite observatory is Sophia. It's actually, I think, really cool. It's a 747 short with a 3-meter dish that they actually just open up the door mid-flight to do spectroscopy above the Earth's water and actually see into the far infrared. But the trick is, we need spectra of these molecules to compare to if we actually want to find these things in space. All right, so how do we do it? Well, some of you may be pretty familiar with this. This is just an RD Pellet Press. We take our sample, we grind it up, and then we press it with just polyethylene powder. It's very transparent in the terahertz, and we make a little puck like this with sample embedded in it. And the idea is hopefully to get some kind of temperature-dependent spectra out. And so then this is one other thing that's really cool in our lab. The way we make our light, one of the few good ways of getting into the terahertz is we actually take an ultra-fast laser, about 60 gigawatts of peak power, we focus it down to a point, and we make a small little plasma filament in the lab. So we get this tiny little plasma filament. We ionize it so completely, we take all the electrons off, and we get this little plasma that throws off this beautiful rainbow of color. And it also gives off a very short, very broad band, so about 8 terahertz wide, pulse of terahertz light. So actually we have to block the rainbow. It's kind of sad, but it's not useful for our spectroscopy. So we get this plasma, we set our sample in the way, and then we get a short terahertz pulse out. Now this is in time domain, so not frequency domain, and it's only a few picoseconds wide, but it covers our entire bandwidth at once. So this would be like doing your entire spectra. You're taking your entire spectra at once. We pass it through the sample, it gets somewhat absorbed, and we detect it, and then we just take a Fourier transform of that, and we have our spectra. So we do sample, reference, and then we take the ratio between those, and we get a nice absorption spectra. So to start off with, what we wanted to do was start fairly simple. A 2, 3, and 4-membered PAH, no functionalization, nothing crazy, just 3 simple systems to test this out. They're all crystalline polyethylene substrate, and as Marco was explaining, the subtle shifts in the spectra caused by temperature variations, and if we actually really want to get the spectroscopy right and identify these things well in space, we need to do this at a lot of temperatures. So what we do is we place the samples in a cold head and just tune the temperature and take spectra at each individual temperature. So we went and did this. So first, this is our spectra for naphthalene. So this right here is actually a polyethylene resonance we can't get rid of. It's a little, well, we can't get rid of it, but for this run, it was actually kind of rough. So going back to the question that was asked earlier, if you want to do theory on these things, simple harmonic vibrational frequency calculations, you'd think, oh, maybe I can sort of work out what some of the vibrations are, identify what we should see in the spectra ahead of time. But it turns out if you do that calculation, what you get are 2 modes right here and right here, one of which we might see, but there's, I should say, actually, this is transmittance, not absorption, and it got cut off. So my peaks point down, not up like Marco's did. So we have strong absorption here, here, and here that are completely missed by theory. But this is actually nice because we get several very strong absorption features. So going down to 0 is basically completely opaque. So several strong, unique absorption features for naphthalene. Moving on to anthracene, same thing. We got 2 very nice strong absorption features and maybe one more over here. Theory says we should have 3. This one may just be a sensitivity issue with our instrument, but these 2 may actually be the same 2. It's just that they're so anharmonic. The theory gets them very, very wrong. And then for pyrene, this is where we were really excited because we got a ton of absorption features all the way across our spectra that are completely different from any of the other PAHs we studied. And if you ask what the 2 theory says we should get 2. So there's a lot of features going on here that we didn't expect to see. We're very excited about. So that's it. We have our spectra. So sort of take-home messages for this are, well, the PAH spectra are unique and they're very promising. Crystalline samples are pretty useful. And the big thing is simple ab initio is okay, but it's a guidepost at best for what you're going to see. And so actually going forward, we actually have applied for time with Sophia to go look for these. So do preliminary proof of concept observations to see if this can be done at all. And then obviously we want to move the lab spectra onto more interesting species. So bigger PAHs that are more relevant for astrophysics and more functionalized systems, things with OH bonds, nitrogen inserted into the ring to start building up a library of more diverse PAHs that we expect to see in space. All right, with that, I'd like to thank my group, our funding agencies, and you for your attention.
Questions for Brandon?
What are those?
Hold on a second.
Oh, yeah.
This is webcast.
Gotcha. So I'm just curious, do you know what those abiotic membranes are actually composed of?
It's a mix of things. So that's actually, the big suggestion isn't that they're just PAHs. There's a mix of there's some short-chain fatty acids that are in there. So actually the big suggestion is that maybe the fatty acids are doing a lot of the heavy lifting in terms of self-assembly, but you incorporate functionalized PAHs because they're there in reasonable abundance, and it might actually look something like what cholesterol looks like in a cell membrane now. And that if you start inserting that, you can do this experiment. If you insert them into the cell membrane, you actually mess with the porosity quite a bit. So it actually might tune it pretty well to act as a very primitive cell membrane.
One more really quick question? Yes? No? All right. Let's thank Brandon again.